this new method of spheroid cultures can be used to study the tumor heterogeneity, to analyze the presence of different populations of cancer stem cells, and also can be nicely applied to for through-output screen for new drugs.
This is a robust and reproducible culture system for generating large number of homogeneously sized spheroids. Moreover, these spheroids can be used to study cancer stem cells. This method is set up to study colon cancer biology, but it can be easily set up to study other cancers by changing the 3D culture conditions.
Be sure to watch for bubbles in the medium, to not move the plate too much during the spheroid formation, and to be careful when harvesting the spheroids into the strainer.
Begin by pre-treating the wells of a 24-well plate with 500 microliters of anti-adherence rinsing solution per well. After a few seconds, centrifuge the plate in a swinging bucket rotor and rinse each well with 2 milliliters of warm basil medium. Observe the plate under the microscope to ensure that the bubbles have been completely removed from the microwells. If no bubbles are observed, rinse the wells again before adding 1 milliliter of warm DMEM basement membrane matrix medium to each well. To induce spheroid formation, seed the desired concentration of CACO2 cells in a 2D monolayer in a 10 cm dish in DME.